Asthma is a disease of the respiratory system. Unfortunately, it is not very rare. According to the World Health Organization, there were over 339 million people in the world that had asthma in 2016. Moreover, 417,918 people are estimated to have died from this disease in 2016. Therefore, the treatment of this disease is crucial since it attacks the human respiratory system and thus it can be deadly. Over the past centuries, the ways to treat asthma have considerably evolved. In order to better understand the modern medical treatments of this disease, let us first understand its mechanism of action. As we can observe here, in a normal human respiratory system, the exchange of oxygen and carbon dioxide is continuous and equal. The smooth muscles that are present along the airways are relaxed and the lining is thin. This allows the air to flow in and out of the respiratory system such that enough oxygen is supplied. During an asthma attack, the smooth muscles contract tightly and the lining thickens such that the airways become swollen. Excess mucus is also secreted, which contributes to partially blocking the airways. This prevents enough air to enter the respiratory system, thus forcing carbon dioxide-rich air to remain in the lungs for a longer period of time. These attacks are responsive triggers, which are very different from one person to the other. Examples of triggers could be allergies, smoke, infection, medication, volatile irritators, etc. The symptoms of an asthma attack vary, but the most common ones are shortness of breath, panic, and cough. The human body has several mechanisms to deal with diseases and viruses, remodeling being one of them. Dr. Domenico Spina, researcher at Ghent University, defines remodeling to be an alteration in size, mass, or number of tissue structural components that occurs during growth or in response to injury and/or inflammation. In other words, remodeling is a normal mechanism of the human body to develop and/or repair injuries, which normally cause inflammation, as white blood cells intervene to repair the damage caused. However, in the case of an asthma attack, this mechanism becomes malfunctioning. According to Dr. Spina, it is associated with abnormally altered tissue structure and function. Asthma attacks are characterized by an increase in the production of white blood cells by the human body, namely mast cells, lymphocytes, and eosinophils. These, in turn, release a protein named cytokine. During the attack, inflammation is caused by the invasion of inflammatory cells in the bronchiole and alveolar walls. Eosinophils can produce inflammatory toxins as well as cytokines. Cytokines also augment the ability of eosinophils to occupy the tissues, which aggravates the inflammation. This is characteristic of milder forms of asthma, whereas in the more severe cases, the infiltration is done by neutrophils, another type of white blood cells rather than eosinophils. Here is a short animation that shows the mechanism of an asthma attack. Here we can clearly see the airways thickening, the muscles contracting, and the mucus being secreted, which prevents the air from properly going into the lungs. The most widespread form of treatment of this disease are inhalers. These are devices which allow for medication to be directly sprayed in one's mouth, thus having direct access to the respiratory system. Other methods, such as oral or injections, are also available. However, these prove to be less efficient since the medication needs to make its way to the lungs before relieving the respiratory system, whereas it is directly accessible when absorbed with an inhaler. This form of medication is often a combination of chemical compounds, the most important ones being corticosteroids and bronchodilators. According to the National Cancer Institute, corticosteroids are steroid hormones which are naturally secreted in the body but can be made in laboratories in order to be used as medication. These hormones have an important role in the treatment of asthma since they block many of the inflammatory pathways that are abnormally activated in asthma, and they have a wide spectrum of anti-inflammatory actions. Corticosteroids will attach to glucocorticoid receptors which are present in human body cells and therefore activate them. These receptors participate in the regulation of gene transcription. During the transcription process in the formation of inflammatory cells and adhesion molecules, which are implicated in the transportation of inflammatory cells, there are interaction with the glucocorticoid receptors. This way, the deacetylation of histone proteins, i.e. the removal of the methyl group from these proteins, is triggered. This causes the DNA strands to strongly associate with the histones such that they become unavailable for transcription. Therefore, the production of inflammatory cells is reduced while the production of anti-inflammatory compounds can be increased. By preventing the synthesis of cytokine, for instance, corticosteroids lead to programmed cell death of eosinophils since these cells are dependent on the presence of cytokines. In addition, they hinder the production of mucus in the airways by affecting the submucosal glands. In essence, by inhibiting the transcription process of inflammatory compounds, corticosteroids trigger a domino effect which prevents inflammation in the airways of the human respiratory system. The most important forms of inhaled corticosteroids, also known as glucocorticoids, are the following. These compounds present some similarities in their structures, namely the four-ring steroid backbone. All of the compounds also have two double bonds within the first ring between carbons 1 and 2, as well as carbons 4 and 5. Besides, they all have an oxygen atom double bonded to carbon 3, a hydroxide group bonded to carbon 11, and a ketone group bonded to carbon 17, although not the same ketone group in each compound. Additionally, a study published in the scientific journal Cell Research observed the chemical structures of different corticosteroid compounds in order to establish a correlation between chemical structures and efficiency of the medication. This experiment compared three different corticosteroids, cortisol, DEX, and MF. As a result, it was observed that the presence of two double bonds in the first ring enhances the efficiency of the medication as it strengthens the hydrogen bond formed during the medication's interaction with amino acids in the human body. A single bond, such as in cortisol, offers too much flexibility in the bond and therefore requires additional strengthening of the hydrogen bond. Besides, it was observed that the C17 alpha position is a very important one when it comes to the reactivity of the compound. Lipophilic ester groups were determined to be preferable in order to boost efficiency. For example, such a group can be observed in the structure of fluticasone propionate. As defined by the National Cancer Institute, bronchodilators are a type of drug that causes small airways in the lungs to open up. Bronchodilators are inhaled and are used to treat breathing disorders, such as asthma or emphysema. There are two categories of this drug, short-acting and long-acting. Furthermore, there are several types of bronchodilators. Beta-agonists and anticholinergics are the two that are the most used for the treatment of asthma with inhalers. There is another type, theophylline, which can be used to treat asthma orally. However, its mechanism is less familiar to scientists, and some disagree to classify it as a bronchodilator medication. Its use has also declined with the years as it is not considered the most effective form of drug for asthma. Beta agonists are used mainly to relieve excess muscle contraction in the airways. However, they also have the ability to hinder the activity of some pulmonary cells, namely mast cells. These compounds interact with glycoproteins and receptors present on cells in the human body in order to stabilize the receptor in its active conformation. This allows the body to undergo a process of homologous desensitization. Desensitization is a process by which the beta agonists occupy receptors, leading to the relaxation of muscles along the airways as the drug reduces the body cell's reaction to stimuli. In addition, beta agonists also have the ability to affect lymphocytes receptors, which triggers a domino effect and reduces the production of inflammatory cells. The most common forms of beta agonists used in the treatment of asthma are the following. It is to be noted that adrenaline is also called epinephrine, and that salbutamol is also known as albuterol. According to Dr. Spina, the majority of the beta-2 adrenoceptor agonists currently used in asthma are derived from the known structure of adrenaline and share a phenylethylamine structure with a catechol, resorcinol, or related moiety that confers potency and an ethynylamine side chain, which confers selectivity to the molecule. Therefore, the different groups on each compound give each one their unique attributes. For instance, the long-acting characteristic of salmeterol is attributed to its nonpolar substituent attached to the nitrogen atom. Besides, adrenaline, isoprenaline, phenoterol, and formoterol are better desensitizing agents due to their superior efficiency to stabilize the receptor. The second category of most commonly used bronchodilators are anticholinergics. Most of the nerves present in the human respiratory system are cholinergic since they release acetylcholine as a signal to stimulate muscle contraction in the airways and secretion of mucus. The triggers that cause asthma attacks can enhance this mechanism through neural stimulation. Anticholinergics attack the muscarinic receptors in order to prevent acetylcholine from being produced, thus preventing smooth muscle contraction in the airways. However, there are three different types of receptors in the respiratory system which have different functions. The current anticholinergics used to treat asthma do not have the ability to act selectively, which hinders their efficiency. For this reason, scientists are trying to produce anticholinergics synthetically in order to provide a better relief of the respiratory system. The most common anticholinergics used in the treatment of asthma are the following. All of these compound structures have a common ester group with different R groups attached on both ends. The ring structure is similar. However, the tiotropium is the only compound with a double bond within the six-membered ring. Overall, these three types of drugs used in inhalers have different functions and different structures. For this reason, their combined use is possible and their properties are complementary. According to Professor Peter J. Barnes, corticosteroids increase the gene transcription of beta-2 receptors, resulting in increased expression of cell surface receptors. In this way, corticosteroids protect against the downregulation of beta-2 receptors after long-term administration. This may be important for the non-bronchodilator effects of beta-2 agonists, such as mast cell stabilization. Therefore, the coupled use of corticosteroids, beta-agonists, and anticholinergics allows for the relief of all symptoms of asthma attacks and are most effectively assimilated by the human body under the form of inhalers.
Hormones can be described as a product of a living cell circulating in the body in order to produce a stimulatory effect in cells. Therefore, in other words, hormones are chemical messengers that allow for communication between organs and other parts of the body so that their respective functions can be accomplished. There are many types of hormones. They have different purposes such as development and growth, metabolism of food, sexual function and reproductive growth and health, cognitive function and mood, and maintenance of body temperature and thirst. Hormones can be polypeptides, amines, terpenoids, steroids, or phenolic compounds. Hormones are a part of a molecule, so some can have much effect on the body. Here are one or two examples of types of hormones. There are growth hormones, such as steroid hormones in the form of glucocorticoids. There are metabolism hormones, such as insulin. There are sexual hormones, such as estrogen and testosterone. There are mood hormones, such as dopamine and serotonin. There are also body temperature regulation hormones, which are called thyroid hormones. Like the others, thyroid hormones are very important, and their function is to regulate the rate at which calories are burned, affecting weight loss or weight gain. This can slow down or speed up the heartbeat. It is important to note that hormones are different from neurotransmitters. By definition, hormones are chemical messengers that allow for communication between tissue or an organ that is far from the starting point. They are produced by endocrine glands and are secreted in the blood in animals and sap in plants to be transported to the site of action. On the other hand, neurotransmitters are a part of the nervous system, hence they are only found in animals. Neurotransmitters are made by presynaptic neurons and are secreted into the synapse. They have an effect on target cells which are in direct opposition to the synapse. And so, the main difference between hormones and neurotransmitters is their site of action and site of release. Epinephrine's primary functions are to increase cardiac output, volume of blood being pumped by the heart, and to raise glucose levels in the blood. It is usually secreted during intense stress, and its stimulatory effects fortify and prepare an individual to respond to a perceived harmful attack or threat in order to survive. The actions of epinephrine are complex because of its stimulatory effects on the alpha and beta adrenergic receptors that produce many responses, depending on the specific receptor and the tissue in which it occurs. Historically, in the late 1800s, epinephrine was discovered. George Oliver and Sir Edward Albert were English physiologists that first described the blood pressure raising effect on a substance from the adrenal medulla. Then, by 1900, epinephrine had been isolated and identified by American physiological chemist John Jacob Abel, and as well as independently by Japanese-American biochemist Jukishi Takamine. It was in 1904 that German chemist Friedrich Stolz became the first to synthesize the hormone. Epinephrine is present in EpiPens. EpiPens are autoinjectors of epinephrine and they were first invented by Sheldon Kaplan in the 70s. They are used in emergencies to treat very serious allergic reactions to insect stings, bites, foods, drugs, or other substances. Epinephrine acts quickly to improve breathing, stimulate the heart, raise a dropping blood pressure, reverse hives, and reduce swelling in the face, lips, and throat. EpiPens contain a dose of 0.3 milligrams of epinephrine.
Epinephrine is released from the adrenal medulla, inner part of the adrenal gland, and glucocorticoids from the adrenal cortex, which initiates the biological response, permitting the organ to cope with adverse physiological, psychological, and environmental stressors. Following its massive release during stress, epinephrine must be restored to replenish cellular pools and sustain release to maintain heightened awareness and sequelae responses in order to re-establish homeostasis and ensure survival. Epinephrine is regulated in part through its biosynthesis catalyzed by the phenyl enzyme in the catecholamine pathway called phenylethanolamine. This in turn is controlled through hormonal and neural stimuli which exert their effects on gene transcription through protein stability. As seen on screen, the pathway first consists of the phenylalanine molecule turning into tyrosine, tidopa, then to dopamine, which turns into norepinephrine, and finally into epinephrine itself. Racemic adrenaline is a mixture of dextro-D and levo-L rotary isomers of adrenaline, where it is the L-adrenaline which is in the active form. Therefore, the racemic mixture is less active and may have fewer cardiovascular side effects when nebulized. Synthetic epinephrine is synthesized in labs through a complex mechanism. First, pyrocatechol and chloroacetyl chloride are mixed to react with phosphoryl chloride and HCl, which in turn creates chloroacetophenone. Then it reacts with methylamine and HCl, creating adrenaline. And finally, after reacting with two H pluses, we obtain adrenaline, or in other words, epinephrine. High-performance liquid chromatography, or in other words HPLC, is a form of column chromatography that is often used for enantiomeric separations of epinephrine. This is possible on the acylated beta-cyclodextrin column. Tyrosine, dopamine, and epinephrine are baseline resolved by a combination of reverse phase, ion exchange, and ion exclusion mechanisms. Chiral capillary electrophoresis is an established analytical technique for chiral separations. All in all, the enantiomeric separation of adrenaline, or epinephrine, and its analogs has been developed using beta-cyclodextrins and the chiral selectors. And the difference in interaction energy for the side chain part is most likely responsible for the enantiomeric separation. For enantiomeric purity assessment, we can use chiral columns or mobile phase modifiers to separate enantiomers before detection. Adding a circular dichirism detector to an achiral HPLC method from the US Pharmacopoeia is shown to be amenable for the determination of the enantiomeric purity of epinephrine. Similarly, achiral chromatography performed using a USP method with a circular dichirism detection may serve as a general means of determining chiral drug enantiomer purity and avoid the need for the development of additional chiral-specific methods for each individual drug.
Synthesis of albuterol. Albuterol is synthesized in many different ways. However, the following is a way better understood by Organic Chemistry 1 students. This is by no means the most efficient way to synthesize albuterol, but it is nonetheless one of the methods. Now for the starting compound, we have a hydroxy group, which is an orthopara director, and two oxygens that act as meta-directors. Therefore, the new attaching group needs to be either ortho to the hydroxy group or para. And because of steric hindrance, we can achieve the target, which is the para position. The first step is acylation, and with the new added chain, now we have a leaving group at the end of the chain, which can then do the second step, which is the nucleophilic reaction. The H plus takes away the bromine, and with the leaving group gone, the electron deficient carbon attracts the nitrogen, which is now negatively charged, and the following group is formed. And with the third reaction, the oxygens without a hydrogen attached to them now gain a hydrogen, and the ether is now reduced, which leaves a hydroxy group attached to a carbon, and which is then attached to the benzene ring. Now the final reaction reduces and removes the phenyl group and leaves the nitrogen with hydrogen, which is used in the body and will be explained later. Beta agonists such as epinephrine, albuterol, and many others have only one important chiral center that determines whether or not the drug is efficient as a bronchodilator, non-reactive, or downright dangerous. We will explain each of the isomers in future slides. Figures shown, it is very obvious that bronchodilators need to have a specific structure or they won't react with the three chains responsible for relaxing the smooth muscles. Now, for a bronchodilator to be extremely efficient, it needs to react with all three chains simultaneously, or it will have different consequences. In S-albuterol, the hydroxy group on the chiral center is not positioned well for this configuration to be a good bronchodilator. Now, when this happens, either only the two other parts of the bronchodilator react with the two other proteins, or the only one of them reacts and throws off the two other ends of the bronchodilator so they don't react with the two other proteins. Either way, it either is extremely inefficient as a bronchodilator, or in the case of S-albuterol, it causes many side effects such as a racing heart, which is called tachycardia. It happens when the heart rate goes above 100 beats per minute, or muscle cramps or other side effects explained by Meriem in earlier slides. Contrary to S-albuterol, R-albuterol assures that all the key components of the bronchodilator react with the three different protein chains in the body and give the desired effect. Now, as it goes, the hydroxy group on the chiral center is R in most of the bronchodilators because this is the desired position to make sure that the key components react with the right proteins. In this presentation, we have talked about bronchodilators. The two main bronchodilators used today are epinephrine and albuterol. They're both very similar in their structure, but they have different uses. Their structures are similar in the sense that they both have hydroxy groups around the same positions. They both have a benzene ring, and they both have a nitrogen with a functional group attached to it away from the benzene ring. We'll talk about the differences in future slides. One of the differences between the two is a hydroxy group that's distanced from the ring by an extra carbon. Now, this extra carbon at first might not seem very important, but it is extremely crucial to the function of albuterol, and it creates a great distance between the two and their functions. So, this extra carbon, as I said before, creates a distance between the oxygen and the ring. This is important because, as we learned from the previous slides, the oxygen needs the electrons around itself to form hydrogen bonds with the proteins. Now, when the oxygen is closer to the ring, the electrons in a pi bond can take part in resonance with the ring and activate it, thereby making hydrogen bond forming extremely slow. By distancing the oxygen from the ring, the biochemists ensure that the reaction happens as fast as possible and the hydrogen bonds are formed as quickly as possible. In the previous slide, we discussed how having an extra carbon between the hydroxy group and the ring creates different rates between the reactions of the hydroxy group in albuterol and adrenaline. Now, in this slide, we will discuss how having different functional groups attached to the nitrogen will affect the speed and the differing uses of different bronchodilators. It is apparent that having bulkier groups attached to the nitrogen will add steric hindrance, which then slows down the reactions between the nitrogen and the proteins in the body. So, when chemists add bigger and bulkier groups to the nitrogen, they create more over-the-counter drugs or long-acting agonists. These are drugs that take longer for their effects to take place, so they are safer and day-to-day use won't affect your body's tolerance to stronger drugs such as albuterol. Now, albuterol and adrenaline are short-acting beta agonists, and this is because they don't have the steric hindrance that long-acting agonists do. Albuterol, however, is faster than adrenaline and is more widely used in emergencies and extreme situations. You might say that albuterol has more steric hindrance because of tert-butyl, but the fact that it has more carbons means that the nitrogen will draw extra electrons from the carbons because of the differing electronegativity. Tert-butyl is the smallest, bulkiest group that can be attached to the nitrogen to make sure that nitrogen is the most basic while maintaining its least bulky state. Because of that, albuterol is extremely effective, and if used on a day-to-day basis, it can create a tolerance in the body which is detrimental to people who have either allergies or need asthma relief.
Today, the most widespread form of treating asthma is with the use of inhalers, often composed of a combination of corticosteroids and bronchodilators, mainly beta agonists and anticholinergics. This is due to the fact that these compounds, due to their unique molecular structures, tackle different symptoms of the disease and work together to relieve the respiratory system. As previously explained, corticosteroids are effective in the inhibition of the formation of inflammatory cells since it directly intervenes in the process of cell DNA transcription by activating glucocorticoids, which will interact with transcription factors and render DNA inaccessible for transcription. On the other hand, beta agonists will mostly stabilize receptors, called beta-2 receptors, which control smooth muscle contraction in order to reverse this process. This mechanism is known as homologous desensitization. Furthermore, anticholinergics will attack the nerves in the respiratory airways, preventing them from producing acetylcholine, which triggers the contraction of smooth airway muscles and the secretion of mucus. Two important beta-agonist compounds mentioned in this video and often used in drugs are adrenaline, also called epinephrine, and salbutamol, also known as albuterol. As we previously discussed, these drugs are synthetically fabricated in order to mimic the natural compounds in the human body. In this way, they are able to effectively interact with proteins, enzymes, and receptors in order to fulfill their role and relieve the respiratory system during asthma attacks. Even though these drugs are efficient in the treatment of asthma, scientists are constantly searching for more efficient forms of medication. Besides, these drugs have many negative side effects. Corticosteroids, for example, can trigger cough, pneumonia, and bruising. Bronchodilators can also be a trigger for cough, but also headaches, nausea, muscle cramps, and palpitation. For this reason, researchers are constantly altering chemical structures to find new derivatives of these medications that can be more efficient and less damaging. According to an article from the scientific journal Medical Clinics of North America, the newest medications are biologically engineered antibodies designed to interfere with discrete steps in the allergic inflammatory cascade. Researchers are therefore working on finding ways to use technology and produce derivatives of known drugs in order to directly act on the affected regions without inhibiting any other normal functions of the human body, triggering such problematic side effects. Another example of such discoveries would be tiotropium bromide, which is an anticholinergic medication synthetically produced that will act selectively on the different types of muscarinic receptors in the respiratory systems to respond more effectively to asthma attacks and other diseases. Now, you understand better the mechanism of action of medication that some people use on a daily basis. Thank you for watching, and we hope you enjoyed the video.